Hello students, this is day 2 and day 3 of AC24, which is our taster gene experiment. Today I'm going to put together both the digestion with the FNU4H1 restriction enzyme as well as the gel. The gel doesn't take too long to explain, and I already explained it on AC23, so if you need some more you can always go back and view that one. So just to recap, what we did yesterday is we used the sterile loop and we scraped some cheek cells out of our own cheeks, and we added some Chelac solution, some proteinase K. We let those both work for a little while. We spun it down, we took out the DNA from the supernatant, and then we ran a overnight PCR. We did 40 cycles, and the PCR steps were also explained in the first video in case you need to go back and watch that. So hopefully in our tubes now, what we have is millions and millions of copies of specifically the taster gene that we're looking for. And so what we're going to do today is we're going to do a digest using the restriction enzyme right here, the FNU4H1. And what that's going to do is that's going to break apart our DNA in a very, very, very specific way. And then we can run the DNA on a gel and it will separate it by size, and we'll be able to tell what genes we have based on what size bands show up. So here's a picture of what the PTC taster gene looks like. So the entire gene that we amplified yesterday with PCR is about 300 base pairs long or so. What a restriction enzyme is it is an enzyme that cuts at a very, very specific sequence in the DNA strand. So in this case, the FNU4H1 cuts whenever it sees a sequence like this. There has to be a G, a C, and then there can be any letter here. This N signifies that there could be any nucleotide. It could be an A, a T, a C, or a G. FNU4H1 doesn't care. It will cut with any letter there. So it needs to be GC, then any letter, and then another GC. And of course the complementary base pairs on the other side. CG, that complement to whatever was up top, and CG at the end. All right, and it doesn't cut exactly straight through the DNA. What it does is it will cut just prior to the N, and it will also cut over on the other side of the N. And what this does is it leaves a little bit of overhang on both of those DNAs. And as we'll do in the pGlow lab a little bit later, we can use that overhang in order to put these pieces back together. That's actually how we would insert a gene into another genome or another strand of DNA, is we would use a restriction enzyme and then do a ligation reaction to put them back together. Right. So ligation is to put back together, and anytime we do a restriction enzyme, that's called a digest. So digest is to cut apart, and ligation is to put back together. So why did we pick this gene? Well, why did we pick this restriction enzyme? Well, it just so happens that there is this sequence in our taster gene right here. It's about 3/4 of the way through the gene or so. So if the FNU4H1 restriction enzyme does cut it, if it cuts the genome, it cuts this 303 base pair gene into two pieces. One of them is about 238 and one of them is about 65. Now, this FNU4HI doesn't cut every single taster gene. It'll only cut taster genes that are dominant, that has a capital T here. So if you taste the paper, you will either be homozygous dominant or heterozygous. Those are taste. You will not taste, do not taste, if you are homozygous recessive. People who are homozygous recessive do not taste the PTC paper. People who are homozygous dominant or heterozygous will taste the PTC paper. People who are dominant or have at least one dominant gene will also be cut. The FNU4H1 enzyme will cut through their genome right at that location. right? Because you'll notice it matches the GCTGC matches the GCNGC that's up top. What's different though, right? So this one is a C. The nucleotide is a C there, and here in the non-taster it's a T. So we only have one nucleotide difference between two different people possibly, and that makes all the difference in the world in terms of whether or not they can taste PTC paper. So the FNU4H1 enzyme will not cut the recessive, not cut, and it will cut the dominant. This will be very important when we go to run this on the gel, right? These results will show up on the gel depending on whether or not it will cut the gene. But in order to get that, we haven't gotten there quite yet. We have to do the digestion first. So we're going to take the PCR tube from the machine. We're going to take 50 microliters, which should be your full volume. You should have added 50 microliters yesterday, so we're going to take all of it out. And we're going to add that to a new 1.5 mil, those are the big white clear tubes, and label it Taster Undig for undigested. All right, so that means we're not going to do a digest. This is going to serve as our control tube for the experiment. It's always nice to have a control tube. And now we're going to label another 1.5 mil microcentrifuge tube and label this one DIG as in yes, we are going to do a digest. Yes. We're going to add the restriction enzyme to this tube, so we will digest it, and we will not add the restriction enzyme to the undigested tube. So to the taster digested tube, right, so remember this is the one that's going to get the enzyme eventually, we're going to add 10 microliters of our digestion reaction mix. This contains a buffer solution, just like all of our other solutions, and it also contains the restriction enzyme, the FNU-H41 restriction enzyme. All right. We are only going to add that to the digest tube. That's the only one that's going to get the enzyme. Okay. So we're going to add in the enzyme first, and then we're going to grab a new tip because we don't want to contaminate anything. And we're going to add 20 microliters of the PCR product from our undig tube that we transferred it to and put it into the digest tube that has 10 microliters of the reaction mix in it. Right? So now this tube should have 30 microliters total. Should have 30 microliters total. It should have 20 of the PCR product and it should have 10 of the digestion reaction mix in it. We're going to place this tube in the 37-degree digital dry bath overnight, and we're going to put the undigested tube into the refrigerator. The restriction enzyme works best at 37 degrees, and so that's why we're going to put it in there. We'll let it warm up overnight and do its thing. So when we come in on day 3, what we're going to do is we'll pour our gel in the normal way. We'll use the large weld combs, so we'll have 6 lanes to run. To the 2 new microcentrifuge tubes that we've labeled, we'll add 15 microliters from the undigested tube from yesterday. So this is yesterday's tube. We're going to take 15 microliters out of yesterday's tube, and we're going to add it to a new tube that we've labeled undig. And we're also going to add 3 microliters of loading dye to that tube. So that tube now should have 18 microliters in it, and we're going to load 10 of that into lane 2. And we're also going to do the same thing with our digest tube. So we're going to label a new tube. We're going to add 15 microliters from yesterday's tube, 3 microliters of loading dye, and we're going to put that into lane 3 for partner number 1. Partner number 2 is going to do the exact same thing and load their gels into lane 4 and 5, and then we will put markers in lane 1 and 6. And again, we're just putting markers in those lanes, both of them, so it's easier to see. We'll turn the gel apparatus on and it will run. And what we're hopefully going to find, what we drew yesterday is, oops, I'm trying to find it. What we drew over here is what we will be able to see, right? So we know that if you are a non-taster, you are a homozygous recessive, so you have 2 little t's, which means that you are in this category, and the taster gene, the taster restriction enzyme, will not cut your genome at all. So you will have 303 base pair sections. Remember, you have 2 alleles of the taster gene, one from mom and one from dad. People who are non-tasters will have 2 303 base pair bands that will show up. These are very heavy, so they'll show up at the top. Right? They're heavier, they're longer than the other ones that we're going to get anyway, so they will show up at the top. If you're a taster, you have 2 options now. Right? You could be homozygous dominant, capital T, capital T. In that case, both of your alleles will be cut, right? Because they're in this camp, and both of them will in fact be cut. So you will get one piece of that's 238, one piece that's 65 for your first allele, and your second allele, since it is the same letter, it will be the same thing. You will also get a 238 and a 65. So on your gel, you will see a piece that's 238, and you'll see a piece that runs down a little bit lower, that's 65. So you will only see 2 bands. So homozygous recessive people will see 1 band. Homozygous dominant people should see 2 bands. Right? And now we do the slightly more complicated one. We'll do the heterozygous individual. We have 1 that cuts, and we have 1 that does not cut in this case, right? So the capital T will give you a 238 and a 65, whereas the small t allele will give you a 303. So this person will see 3 bands. Right? They will see a large 303, they'll see a 238, and they'll see a 65 that runs down low. Whereas the 2 bands and only 1 band for the homozygous recessive taster. All right? So given the fact that there is a restriction enzyme in the gene, that allows this whole experiment to work. If this restriction enzyme was not here, we would have no way to run a gel and see what would work, right? Because every single piece on the gel would show up at 303 because nothing would be able to cut it, so it wouldn't be a good experiment. So when you're planning something like this, you always got to make sure that you're going to be able to find a restriction enzyme somewhere in there and see how far down the gel it will run so you can do gel electrophoresis on it. All right, I hope your experiment works. Good luck.